and I'm probably not introducing them in the order that they'll actually talk, but our first speaker in the group is Dr. Aaron Pilnick, and he is a postdoctoral researcher at the Forest, Fisheries, and Geomatics Sciences School here at University of Florida. And he did his Bachelor's of Science in Biology at Tufts University and his PhD with Josh Patterson here at the University of Florida in interdisciplinary ecology. And he has interests including restoration ecology and aquaculture and marine invertebrate biology. And he's currently based at the Florida Aquarium Center for Conservation, which is in Apollo Beach, Florida, and he has research that focuses on aquaculture of sea urchin grazers for coral reef restoration and coastal sponge enhancement, which is what you're going to be hearing about today. And our other speaker today is Dr. Don Behringer, and he is a professor and graduate coordinator also at the School of Forest Fisheries and Geomatics Sciences at University of Florida, and also with a joint appointment in the Emerging Pathogens Institute. Don did his bachelor's in zoology at the University of Florida and a PhD in ecological sciences in my home state of Virginia at Old Dominion University. And his professional interests are general marine ecology, marine crustacean biology, and marine disease ecology. He literally wrote the book on marine disease ecology and is internationally known for his work on crustacean diseases and other topics. And he's published more than 80 papers and book chapters and teaches also the marine ecological processes class and other classes at UF, which are really important parts of the marine science major here at the University of Florida, and all of this is in just 20 short years since graduating from his PhD. So incredibly accomplished, both of our speakers. And so with that, I will stop sharing and hand the screen over to you all. Thank you both for being here.
Oh, thanks for having us. All right, are we set, Aaron? Aaron and I are going to tag team this. So I think things are shared with me. So if we have a problem, we'll address it. But glad to be here and talk to you about hard bottom and sponges and how we might be able to restore or enhance them, and why you should just care about sponges, because sponges are super cool. So title of our presentation is Sponge Habitat Restoration and Enhancement Along the Nature Coast. And of course, you were introduced to Aaron Pilnick, and Josh Patterson is also heavily involved in this project as a co-PI, but we figured it'd be a little too much to try tag team this. So but Josh is very much involved in this as well. Okay. Let's see. All right, so it's not moving forward. Aaron, do I have?
Yeah, Don, I think you should have remote control access. Yeah.
It's not. Oh, maybe if I use wait. There, now it seems to maybe just took a bit. Okay, so before I dig in too much into it, I want to acknowledge the sponsors that have made this work possible: Florida Sea Grant and the National Fish and Wildlife Foundation. So they both contributed to some of the work that you're going to hear about today. Okay, so our coastal waters are changing. This is probably no surprise. We have loss of critical habitat. We're losing mangroves, salt marshes, seagrasses. Of course, the coral reefs are especially in Florida, are under threat from numerous sources. These aren't meant to be exhaustive lists, but we have marine food webs that are being disrupted by these changes. Fisheries are in decline, and coastal infrastructure, frankly, is at risk with loss of some of these foundational species along our coasts. Some of the sources, right? Again, this isn't meant to be exhaustive, but eutrophication, right, is impacting our coastal environments. Harmful algal blooms, which we'll talk a little bit about today. And of course, acidification, warming, increased storm activities, and hypoxia are all impacting our coastal environment. And there you see is a little bit of hard bottom, what we're going to talk about today, and some sponges that were at the forefront of one of those impacts. So what is hard bottom? So that term gets used for several different types of habitat. Some folks actually refer to coral reefs as hard bottom habitat because the bottom in many cases is hard. But the hard bottom we're talking about today is a shallow nearshore hard bottom habitat. And you see sort of an idyllic picture of hard bottom actually from the Florida Keys there on the right. Kind of looks like almost like a tropical forest underwater. But this kind of hard bottom habitat that we're talking about is typified by flat expanses of calcium carbonate rock with a thin veneer of sediment over top of that. It has abundant fixed, some calcareous, some not, algae, and then it also has a lot of ephemeral drift algae. So at times it'll be lush and thick with algae, and other times it won't be. And it also is typified by a diverse benthic assemblage. So it's got sponges, of course, that we're going to talk about today, but octocorals like you see in this image here, solitary hard corals, and other sessile organisms that really form the three-dimensional habitat. So the hard bottom itself is generally relatively flat. There might be solution holes and things that organisms can live in, but generally the habitat, the structural habitat is provided by the organisms that live there. And really that the sponges in this hard bottom habitat are what dominate the biomass. So those sponges, there are over 60 species of sponges around coastal Florida. You see a few of them pictured here. They support the ecological function of hard bottom. They provide that habitat, as we just mentioned, not only around them, lots of organisms, fishes and invertebrates seek that structure, but around them, under them, you see that little stone crab on the right there, and within their canals. In fact, the only eusocial organism, that is an organism that has a structured society, kind of like bees and termites and ants. The only one in the marine environment actually lives within the canals of sponges. It's a snapping shrimp that has that kind of a sort of caste system. They're also extremely important for nutrient cycling, and they can benefit water quality via their filter feeding, but also through microbial activity. Many of the sponges actually have very large microbial communities within their tissues, and that also affects and can benefit the waters around them. All right. And in fact, I should just mention that in the Florida Keys alone, there's an estimation that when the sponge communities were intact, the sponge communities in Florida Bay, that space between the Florida Keys and the southern tip of Florida, that sponge community could filter the entire contents of Florida Bay in just 3 days. So that's a lot of filtering capacity and a lot of water quality improvement that they were capable of. So sponges particularly are under threat. Eutrophication and hypoxia are a couple of the problems, and that sponge you see on the right there is most likely that death was a consequence of a hypoxic event. Disease outbreaks occur on occasion, but in the Florida Keys, probably the most devastating has been harmful algal blooms. So in Florida Bay, right? So this is where at least the sponge work that I've done in the past and folks in Florida Sea Grant have done has focused in the Florida Keys because the sponge communities down there and the hard bottom is so expansive. So that's where the work started initially. And it really started initially down there because of these cyanobacterial blooms or these Synechococcus species. Oftentimes there'll be multiple species involved in these blooms. But what you're looking at there in that image is from the deck of a boat looking down at hard bottom habitat, and you can see loggerhead sponges. Like that loggerhead sponge I'm highlighting right there is probably about the size of a car tire. So they're really big sponges, and historically Florida Bay was gin clear like that, just absolutely clear. But what happens occasionally, and has happened historically, but seems to be happening with increased frequency down in the Florida Keys, is we'll get these blooms, and the water turns from that gin clear to absolutely pea green. Problem with it turning pea green like that is you think, oh, well, that's lots of food for sponges. Problem is these Synechococcus blooms are also they also produce mucilage. So basically, it's a polysaccharide. The water becomes more viscous. And so for something that's filtering water, you don't want to have to try and filter something the consistency of like a witch's brew or like honey, right? So what happens is those sponges end up suffocating and dying. So you might go down, be snorkeling around Florida Bay, which is an average depth of about 1 meter. So anywhere between 1 meter to 10 meters, you're snorkeling around, you see sponges and they look beautiful like this. A bloom comes along in the order of just a few weeks. you go out there and the sponges look like this. And it just devastated the sponges in the Florida Keys. So that has consequences, right? Because sponges have economic importance beyond just being ecologically important for lots of different reasons. They're also economically important. Of course, they are fished themselves because there is still a commercial sponge fishery off the Nature Coast and also down in the Florida Keys. And this image is actually from the Florida Keys. And those are all dried and processed sponges on the roof of that ship there. Historically, going way back, sponges were harvested. I mean, that was the only kind of sponge you could get. But I think it was in the 1930s that artificial sponges were created, and that really took a lot of the pressure off the sponge communities. And largely, those sponges are still harvested, but not at the same rate they were, and they're used for tourists, but also for medical purposes and cosmetics and some of the things like that. So they are still harvested. But of course, as we noted before, sponges are really important for the organisms that live around them, under them, in them, many of which are important for recreational and commercial fisheries. So lots of fishes like groupers and snappers will reside in hard bottom, especially as juveniles. But stone crabs and lobsters also use those sponges. In fact, sponges are the primary habitat used by stone crabs and lobsters, depending on where you are around the state. So they're extremely important, and their loss has ramifications for the economies in the areas where they exist. There's also the potential that, and this is work that was done a number of years ago that suggests that even though in the Florida Keys, the sponges really can't handle these heavy cyanobacteria blooms, that there is some evidence to suggest they might actually be effective to some degree at consuming and reducing red tide events. This is some early work, and additional work needs to be done to show this more equivocally, or unequivocally, I should say, and look at the amount they could actually clear. But it's encouraging. All right, so. Oh, there we go. So that brings us to the idea of restoration enhancement. I think we've made the case, right, that sponges are important. In some places they have been lost. So there is the need, the desire to potentially restore those sponges in places where they're lost. But because they are of the benefits that can be derived from sponges, there's also the possibility of enhancing those communities. So adding sponges to places that maybe there weren't that many sponges, or adding sponges to places where they weren't. And so how do we do that? How is sponge restoration accomplished? And this nice infographic you see on the right here was developed by Florida Sea Grant, which kind of gives you the lowdown. It's not high-tech restoration work. What you're looking at there are little concrete bricks, and those little pieces attached to each one of those little bricks in this image are sponges. And those are little just electrical zip ties holding those sponges down to those bricks. The nice thing about sponges that makes them amenable to this type of restoration is that sponges are only at the cellular level of organization. So that means that you can take a sponge and you can cut it up like a birthday cake, and you can take little pieces of sponge, attach them to a brick like this, and they will heal over and they will attach themselves to that brick, and you have a little experimental unit, essentially a clone of that original sponge. And it works really well. Some species are more amenable to it than others. And so we have to test those sponges to figure out which ones are amenable to it and which ones aren't. So that's sort of the nuts and bolts of it. But this brings us to a really important point, is in order to scale up restoration or enhancement, you really have to understand the ecology of the system, the ecology of restoration before you start that. And so that's a lot of what I'm gonna talk about, and then sort of kick it over to Aaron to expand upon. Because if you don't do that kind of work, you end up with things like this. Is anybody familiar with, or you probably are familiar with, the tire reefs down in Broward County? So I worked as a natural resource specialist for Broward County, right when that could be me in that picture, I don't know. But basically what happened down in Broward County is they had the idea back in the 1970s that they could take car tires, and they could band them together and they would sink them to the bottom, and they would all get bound together, and the corals would grow on them, and we would have these beautiful artificial reefs. They didn't do their due diligence when it came to this and realized that when you take stacks of car tires and you band them together with metal bands and you put them in the salt water, well, what's going to happen is those bands are going to rust and they're going to pop. And the fact that car tires are made from petroleum products, not a lot of things like to grow on them. And so this is they're removing those tires from those reefs now, but this is just to give you an idea of the impact, an extreme example of the impact that not doing your due diligence on a restoration project can have. So that's really our goal, to understand sort of the best practices when approaching sponge restoration. So not only do we not have a catastrophe, but we also improve the likelihood that we have success in what we do, and then that hopefully will encourage and facilitate upscaling and future restoration. Come on, advancing again. Oh, there we go. All right. So some of the considerations in advance of scaling up restoration are things like species selection. So as I mentioned before, not all species are necessarily amenable to this type of restoration. So which species are? Are there interactions between species? Ecologically, some species get along with the others of their species and get along with other species, and others don't. And so we need to know that in order to know how best to approach restoration. How do we know when we've achieved ecological function? Say we're restoring sponge habitat that's been lost. How do we know when we've basically brought that habitat back to its original ecological function? What about some of the abiotic factors to consider, like hypoxia? How do we approach that in areas that might be subject to that? Where do we put sponges? How close to shore can we put them? What about the impact of freshwater? If you put them along a coastline that there might be an inlet nearby, what impact might that have? How deep can the sponges go? What about latitudinal gradients? So there's lots of different things that need to be considered, and we're sort of tackling these one by one as we go along. So we got some funding from Florida Sea Grant and from the National Fish and Wildlife Foundation back it started back in 2021, and we started the permitting process to do this work. And then we started an initial pilot project in the fall of 2022. And we did this down in Hernando County, where we had lots of support and some good foundational information on the hard bottom habitat out there. I mean, the initial goal, it was a small amount of funding, but welcome nonetheless, was to identify candidate species. So what species? We knew some of the species existed in the Florida Keys, also existed off of Hernando County, but whether they would still be amenable to restoration in that habitat is what we needed to know. So we were looking to identify the candidate species, identify donor habitats, identify nursery habitat sites, and then basically setting up those sponges. And we did this at a series of 4 sites, approximately 4 miles from shore, in about 8 to 10 feet depth of water. So we wanted to find areas where we could have nursery habitat that was close to that donor habitat. And I should mention, when I say donor habitat, the nice thing again about sponges being at that cellular level of organization is that we can harvest sponges from a donor site. And as long as we leave some of that sponge, we cut them off and don't rip them off and leave some sponge on the bottom, that sponge regrows. And so you can actually restore enhanced sponges without actually taking them from one place and just putting them someplace else. Okay, so these are the sponge. We did some initial surveys, and we worked with the Hernando Department of Waterworks to find some from donor sites and nursery sites, and we selected this suite of sponges. So loggerhead sponges are, again, they're those they can be large. They start small, but they can be up to the size of a small car. I've seen them, but typically about car-sized tire is not unusual. So the loggerhead sponge doesn't have commercial value as a sponge species, as a harvested sponge species, but it's very important and very abundant from a structural standpoint and for use by lots of organisms. The vase sponge similarly doesn't have commercial value as a harvested species, but is very common and dominant numbers wise. And then this sponge you see in this image here, I didn't have a great picture of it by itself. The purple one there is the branching tube sponge, Iolocroia crassa. Again, those top three are ecologically important, but then we also wanted to include a couple of species that are both ecologically important, but also economically important to the commercial sponge fishery. So the sheepswool sponge on the left and the yellow sponge, Spongia barbara, on the right. So those are the 5 species we targeted initially. And we did this again. Here's an image you see of the area of Florida that we were working in. And then a zoomed in image, you can see Hernando Beach is down here near the bottom of this image. And these are where the sites that we found. Again, initially we wanted to set them up relatively close to one another so that it was for the logistical purposes in Hernando there. There we go. So we initially tried to put out about 250, 200 to 250 of each one of these species. This started in November of 2022. We ended up with 199 loggerhead cuttings deployed, 213 of the purple branching sponges deployed, 236 sheepswool, 225 vase sponges, and only 84 yellow sponge cuttings. And we left them out there. We checked on them at about 6 months and looked at their survivorship after that point in time. And then about a total of 16 months later, in the March of 2024, we did a final assessment to look at their survival. And you can see the survival of those first 3 was really high. I mean, the sheepswool sponges, almost 100% of those sponges survived. Purple also responded extremely well. That's a species that to my knowledge doesn't exist in the Florida Keys, that we hadn't worked with in the past, but survived really well. Loggerhead sponges had decent survival as well. The vase sponges, surprisingly, not so well. They did well in the Florida Keys, but we did have a fair amount of mortality, and why that is, we're hoping to figure out, and Aaron will talk a bit about that in a minute. In this image here of myself and Doug Marcinek, those are vase sponges. So you can see they actually, they look pretty good there, but we lost a fair amount. And lastly was the yellow sponges. The yellow sponges was a bit of a surprise in that they survived quite well in the Florida Keys, but they did not survive well here. And hence the reason why you need to do this kind of work. Had we just assumed that what we had done in the Florida Keys was going to work off the Nature Coast, and folks had scaled up with yellow sponges, it wouldn't have probably worked out so well. And it might be, we only put out 84 cuttings. They were relatively uncommon there. I mean, there some places they were more abundant than others, but it might be that they're at their northern limit to their range. So that was sort of the last that we used those yellow sponges. So you can see we had really high survival. So we had a lot of sponges out there, and all in these little bricks that had healed, these nice little experimental units like you see there. So we said, that project was effectively over, but we had these sponges. So we have these little units. Why don't we do something to try and sort of take this to the next step and investigate some of these ecological questions? So that's what we did. And the next phase of the project, I'll kick it over to Aaron, and he'll tell you a little bit about what we did there and why we did it.
All right, cool. Thanks, Don. I will take over, steer the ship a little bit here. So that was some great context and background information provided. So I think now I'll kind of dive in a little bit and explain, the next phase of the project, and kind of go over how we designed this project, the types of data that we intended to collect, and I'll provide some quick results, and then some outcomes from that. But bear in mind also that this was, data that was collected literally a week and a half ago. So some of the findings that I'll present are really preliminary, and so the data is really fresh. So as I'll mention when we go over that. So all right, so really the goal is, as Don really mentioned, to try to understand some of these ecological factors that are impacting these restoration outcomes. And so with this next phase of this project, the goal was to experimentally try to understand some of these factors through experimentation. So we used these 4 species from the pilot study, that had higher survival. We had to omit the yellow sponge, unfortunately, because the survival to that point was just too low. And then we also aggregated, so there were 5 original sites within that nursery that was set up. And we decided to aggregate some of those sponges and focus efforts on 3 sites. So here's a picture of Brittany Scharf. She's a Florida Sea Grant Extension agent out of Hernando County, who's been helping support this project. So part of the initial setup for the experiment was to bring some of these sponges on board. We actually had a pop-up dog pool and used the live well pump running through there on the UF vessel. So that's kind of, I like that photo. It showed us how we were actually transporting these sponges around between sites. And so for the project design, Don touched on some of these ecological factors earlier, but I'll kind of provide a little bit more detail here. So again, we utilized these established sponge cuttings from the pilot study. There was good survival. We were really interested in 2 factors here. One is the effect of elevation, and I'll talk about that in a second. And then the other is the effect of, or potential effect of intra and inter-specific interactions, which I'll also explain. So let's just jump into the elevation study right here. And so the hypothesis that really was underlying this is that sponges elevated above the benthos would have a higher growth and survival relative to those sponges that were placed closer down to that substrate. And really, there's 2 reasons for our thinking here. One is that there's this hypoxic element where sometimes in this boundary layer, especially following eutrophication events, the sediment will develop a thin layer of hypoxia, and this can essentially affect the sponges. So imagine you have a sponge on a brick and it becomes embedded into that substrate over time. Even if you have a thin layer of hypoxia, that could affect the base or the foundation of the sponge. It may not end up killing the sponge, but it could, potentially cause that base or foundation to decay, and then it can float off or cause other problems. So by elevating the sponge off of the seabed, off of the substrate there, maybe you can reduce some of those impacts just by keeping them distant from any hypoxia. The other thought behind this was that it has to do with the flow dynamics. So closer down towards the substrate, there's more friction, right? So that reduces the water flow. And as these sponges are filter feeding, bacterial plankton, phytoplankton, dissolved organic matter, if they're positioned a little bit higher in the water column, that might increase their access to some of this food supply and their ability to filter feed, which might then result in some improved metrics like growth and survival. And so how do we actually go about doing this? In the top left image there is this is an individual vase sponge, and this is kind of just representative of how we actually elevated them. So they're already attached to those bricks, and then we prop them up on 3 additional bricks just to raise them up off of the bottom. And then so that was our elevation treatment, and then on the bottom is the controller unelevated individual. And this is a little grid just to kind of explain a little bit of the design. So for each treatment, it was represented by all 4 of these species, and these were placed at each of those 3 sites. So for instance, at our first site, we had 6 elevated vase sponges, and those are arranged in a grid where each of those individuals were tagged. And so we knew the exact position and orientation of those individuals so that we could go back and monitor and collect data on those individuals over time. So moving on to the next experiment is the facilitation or inhibition. And so the hypothesis here was that sponges of the same species might enhance the growth and survival when placed right up against each other or paired with each other, while sponges of different species might actually oppositely inhibit growth and survival when paired. So really kind of trying to understand any competitive interactions here where, sponges of the same species might have, might be at an evolutionary disadvantage to inhibit growth and survival of the same species when placed in a really close proximity. So those were that was the hypothesis under for that aspect of the project. And so on the left, again, here's some pictures of how we actually went about doing that. So here's a purple sponge that is placed up against a brick in the top left, and then in the bottom, this is the purple and a sheep's wool sponge. And so the two bricks were literally just joined next to each other, fastened, and then placed in a similar grid structure. You'll notice that, so what's the point of the brick here? So each species was paired with a brick which acted as essentially as a control. How will this species, grow and survive in absence of competition? How will that, a loggerhead how will growth and survival work when it's paired with another loggerhead? And then finally, what does that look like when it's paired with one of the other two species, being purple or sheep's wool? And we reproduce that for each level and for each species essentially. And as mentioned, those were placed in a similar grid structure where we could go back and identify these individually tagged and positioned sponges. So what did our timeline look like? So the project was initiated in June of 2024 after we established, what the project goals were and the grids. We collected some baseline data. So first looking at height. So what is the maximum height from the top of that brick to the top of the sponge? Then we also captured some top-down imagery using this PVC fixed frame with an underwater camera, and I'll talk about in a second, the reason for that. We also recorded sediment depths, trying to understand some of the habitat factors, in addition to some benthic transects to look at benthic composition. And then in August, we followed up after we were able to procure some of these data loggers and get some long-term monitoring of the temperature and salinity as environmental factors. So, okay, so what was how do we actually go about analyzing the growth? So this is kind of a two-part process here. So when we're looking at growth and we're trying to determine, how well their sponges are actually growing, we can look at the vertical growth, and we can also look at the horizontal growth. And essentially, what we're doing here is calculating a specific growth rate, which is just telling us a percent growth per week or per month or however over that 6-month time period. So when we're tracking the same individuals over time, we have a final and an initial size and able to determine basically a percent growth rate. Same thing with the horizontal growth. So here, this is the purpose of that fixed PVC frame, where there's actually a ruler that you can use to slide up and down the frame. And when we position that at the top of the sponge, take the photo, bring that into an image analysis software, we can essentially set a known measurement in there and then draw an outline of the top of the sponge, and then the image analysis will pump out what your circumference in your area is. And then so again, those are two initial and a final growth value that we can use in a specific growth rate calculation to understand what is the percent of horizontal growth per month over a 6-month time period. And then we also wanted to look at survival. And I have survival in quotes here because, for some of these sponges were well attached by the time the study started, but there's a chance that some of them might have become detached and floated off. And we're not really certain if that means that they perished or if they were surviving elsewhere. So it's not really a true survival. What we're trying to understand is the proportion remaining. And so we're able to collect this data by conducting these census over time. But yeah, anyone who has been living in the state of Florida over the past year, especially on the West Coast, might look at this timeline and say, okay, well, I wonder if there were any major storm events that actually happened in the latter half of 2024. And sure enough, we did actually have 3 major storms. First was Debbie in August, which landed north of Hernando Beach there. Helene in September, closely followed by Milton in October. And these were major storm events for the west coast of Florida. And to be completely honest with you, I was sort of expecting mayhem and destruction at our experimental sites, not really knowing what to expect. So in November, we followed up with a post-storm checkup. We wanted to see what the status of our plots were, and actually really good news. So everything looked for the most part like it survived and it weathered this triple threat here. So apart from a few of the elevated, vase sponge in particular that had quite a lot, were a bit higher than some of the other species. Some of those toppled over, and then so we righted those. But for the most part, the grids were all intact, and we were able to move forward with the experiment. So that was a really good outcome right there for us, and interesting also to note. So, all right, so now I'll kind of quickly just breeze through some of our results before I run out of time. So anybody also who's a scuba diver might look at this picture and say, oh, well, why are these guys wearing dry suits here? It's in, 8-10 foot of water in Florida. But as we found out, doing this work here on the West Coast in the early winter months can be quite challenging with water temps in the 50s, 50 Fahrenheit and low 60s. So as we quickly learned, this is dry suit territory, especially as you're spending multiple hours, even at 8 foot, taking photos and recording measurements. It's you need to be well prepared to do that. Okay, so survival data for the elevation. Again, this is just a first pass at some of this data, but this is hopefully will give an indication of the kinds of data that we're collecting and what this might look like in a final pass. So on the y-axis, the percent remaining. On the x-axis, you have your species grouped by treatment. And so really not a whole huge difference in survival between the elevated and the control treatment at the 6-month time point. Vase sponge looks like there was overall a little bit lower survival or percent remaining. Now let's look at the one of the growth metrics. So we're still working on the image analysis for their circumference. That takes quite a lot of time to go through and actually outline each sponge. And we're working with Noah Stouch. If he's tuned in, thanks for your hard work on that. So I really just have the vertical height growth data for right now. So on the y-axis, this is your specific vertical growth rate per month over 6-month time period, broken down by species on the x-axis again. And then the red dotted line will tell you, 0% growth. It means that there was no change over that 6-month period. If it is above that line, it means that there was positive vertical growth, and below, negative vertical growth. So most sponges were growing, except it looks like for the loggerheads, for the elevated loggerheads, for some reason there was actually a decrease in growth, which is really interesting to note. And I don't want to. This is a really noisy graph here, and I don't want to spend too much time on here, but just want to introduce the idea that when you start to look at these individual sites, some of the for instance, loggerhead. The control loggerhead at the third site, N5, was quite a bit lower in its vertical growth, as well as the elevated loggerhead at that site. And so that's really, that could be driving some of these aggregated results that we saw in the prior plot. So what it is indicating is that there's likely these site-specific effects, and what we need to do moving forward is try to tease apart whether they're, the importance of these treatment effects relative to the site effects, and try to understand what are the drivers of these metrics that we're observing over time. So moving on to results for the facilitation inhibition. Again, this is the survival graph. So for the loggerhead group on the left, that first bar is percent remaining when loggerhead is paired with the brick. That's our control. Next bar, loggerhead with itself, loggerhead, so on and so forth. And again, not really looking like there's a huge impact yet of these pairings on the survival. And looking at that vertical growth, we have some box plots again here in the same orientation as the last graph, so broken apart by species. The loggerhead on the left. So that first box plot on the left is the percent vertical growth when the loggerhead is paired with a brick. And any of the green box plots depict positive vertical growth over that 6-month period, and then in red, negative growth. So most things grew vertically, despite, regardless of the pairing, it looks like. We need to dig into this a little bit more. Again, there might have been some competitive interactions with loggerhead. But I just also want to emphasize this is only the vertical growth that we're looking at. So when we are able to, finish the image analysis and look at the circumference in that area, we can fold in some of that horizontal growth, because what might end up be happening is as these sponges, they were establishing on the bricks, maybe, they were sort of encrusting and spreading out onto that base brick. And they might do that for a period of time um, before really growing vertically. So we need to look at more of those growth metrics. And then lastly, I want to quickly look over this environmental data. So here are 3 sites again. Pretty interesting, I think, when we're looking at water temperatures, minimum temps from 7 or 8 degrees Celsius up to a maximum of 32 or 33. That's a pretty large temperature range, averaging around 23. And for salinity also, I think we were kind of surprised to see this. It being still, it's not inshore. I guess you could say it's far enough offshore where you might expect your salinity to not dip down to 11 parts there. So that was interesting to see also. And so the point, I think, is these sponges are really dealing with a wide range of environmental factors, and just of the 2 that we were looking at specifically. This also might, some folks might find this really interesting. So this is data collected straight from the data loggers. And so on the y-axis on the left is temperature and salinity on the right. And so my colors here, I believe that the blue line is your temperature and orange is your salinity. And I think it is, interesting to note, so these vertical red dotted lines are the timelines for those 3 hurricanes that we observed. And if you look at the orange line specifically, looking at the salinity, that first storm, which was Debbie, you see a spike in salinity right around that time point. And the same thing happens with the second storm, which was Helene in September. And so what we think might have been happening there was when these storms have a landfall north of Hernando Beach, they might have been pushing an onshore wind and then driving some of that, more saline water onto shore, and essentially driving that salinity up. Oppositely, for the third storm, which was Milton in October, that landed south of Hernando Beach, and so that was driving an offshore wind. And so if you have a heavy precipitation combined with that offshore wind, you might see this pretty dramatic drop in salinity. And then it comes right back up as you kind of get into the dry season. And then here's the same data for a second site, N1 here. Also, these 2 sites are a little under a quarter mile from each other, but according to this data, if you're looking at the blue line, that's your temperature. So the minimum temperature at site N1 was about 7 or 8 degrees, and then minimum at N5 was a couple degrees above that. So that's kind of interesting. If I return back to the site map here, N5 is the further site of the 2, and so I guess we were sort of expecting almost really similar water quality results or data from these sites given their proximity, but maybe even that quarter mile proximity closer to shore where you might have more freshwater inputs is seemingly to have a little bit of a larger effect than expected. So as I mentioned, next steps in this process, we want to look at the horizontal growth and then start to fold in some of those site-specific factors, such as the environmental data. We want to look at the benthic composition and the sediment depth and try to understand any associations between these factors and the growth and survival metrics that we're recording. And then hopefully, the outcomes from this specific project, we'll be able to hopefully understand what the individual species performance is, what their interactions look like. Maybe we'll hopefully be able to understand a little bit better about some priority habitat, for trying to do this work. And then this will help us to also optimize an outplanting method. So really this will help to kind of advance the restoration methods and capacity through this knowledge. So, another outcome, and as I'm closing here, I wanted to include these 3 videos. So we're seeing, it's a reef, right? These bricks are establishing life here. Stone crabs, pufferfish, porgies. And so really, our big question is what are the long-term or downstream ecological impacts from this restoration work. So we'd like to do for future research to kind of expand this work out. We think we probably need to run it out to 12 or 18 months to start to observe some more noticeable impacts on that growth specifically. We'll have a ton more questions. This is really just kind of Pandora's box. So what is, if we alter the density and the distribution of these sponges, does that affect some of the benthic ecology? Will it increase or minimize localized recruitment? Is there that inshore offshore gradient? How does that impact these restoration outcomes? Are there other species, right? The yellow sponge that works, grows fairly well down in Florida Bay, as Don mentioned, not so well up here, but maybe there are other species of ecological, commercial interest to explore as well. And as mentioned, ultimately the big question and what we'd like to know is what is the return on this restoration. And so longer term, we'd like to study the impact on the benthic communities, fish communities, invertebrate communities. What does the recruitment to these structures look like? Is there a positive effect of actually having that living sponge tissue relative to just bricks out there? What kind of positive ecological function does the living vertical 3-dimensional structure provided by the sponge do? So, and then also what are some impacts on the water quality and plankton communities? So yeah, in closing, we'd like to acknowledge quite a large number of people who have really helped with the project. In the top left, the Josh Patterson Lab. Oh. Doug Marcinek, Noah Stauch in the bottom left for doing a lot of the image analysis. Captain Greg and Brittany Scharf out of Hernando Beach as well, and too many other people to list here. So with that, yeah, I think we'll be able to take some questions here.
Wonderful. Well, you do have lots of questions in the chat. So the first one is, it may be too soon to know, but in the addition to hurricanes, are there impact from other seasonal conditions?
Well, I'll field that one. One of the things that can you hear me? Yeah? Okay.
Yep.
One of the things that we learned early on down in the Florida Keys was that doing this initial sponge restoration setup work in the summertime was not good. Our survivorship was much poorer in the summer when the water was really warm, potentially a little bit more salty. Or just the salinity more variable generally if you have heavy rain events. So from that perspective, it is probably wise to do this kind of work when the water's a little bit cooler. But otherwise, yeah, we haven't seen, other than the hurricane events, a strong seasonal impact. But that's something that hopefully with additional surveys that we're able to accomplish on the sponges that we have out there now, we might be able to see whether that growth rate and that survivorship changes seasonally.
Awesome. The next one is a comment that this is really encouraging and asking about if the red tide blob off the Gulf Coast is a threat to these sponges.
We put up that there is one paper that suggests that sponges can consume Carina brevis. I think I would say it's probably a little bit premature to say that sponges are the cure to red tides, but I think it would be interesting to do some more focused research on different species and their clearance rates that they have, whether they can not only tolerate red tide and tolerate the toxins in the water column, but can they consume them and at what rate? It's tricky research to do because working with Karenia brevis, you have to do it in very controlled laboratory settings. And the problem with sponges is that they don't like being in controlled laboratory settings. It's very difficult to keep sponges happy in a laboratory. So doing that kind of work in the controlled setting that you need to do it in is tough. So I think the jury's out a little bit on that, but I think there's some possibilities there.
Yeah, for sure. And like Don said in the beginning, if it created a big low dissolved oxygen event, that could definitely threaten them. But
Yeah,
that's
indirectly.
cool to know about the direct consumption. This is my question. Are the sponges staying in place at that phase 2 experimental site sort of like for good? Like, is that their outplanting location? And also, are there any sponges left at the nursery site for other research or restoration projects?
You want to field that one, Andrew? You want,
Sure.
or Aaron, you want me to? Go ahead.
Yeah, yeah, I can. Yeah, so I mean, as of right now, the plan, which is really dependent on funding, would be to run the current experiment out 12 and then 18 months. There are sponges that are still there that weren't utilized for the phase 1 experiment that we could incorporate for an additional experiment. Plus, there's the option to establish more cuttings and fold those new cuttings into a new experiment where we can look at some of those other factors. But beyond that, yeah, I'm not sure. Yeah.
As far as where they're going to be left, so in our permitting process, we permitted the sites that we are using now, and that permit basically said that empty bricks where sponges died, those would be removed, but the sponges, living sponges, would be left because eventually the sponges will grow over those bricks and attach to the bottom. And so those bricks are really largely viewed as sort of a de minimis impact on the environment. Although I know in the Florida Keys, there's been a pivot to focus on trying to use natural limestone rocks and things. And so I think that would be something that would need to be considered upon scaling this work up, potentially. It's more difficult and such, but it's something that would probably need to be considered. But the sponges that are out there now are there to stay, the ones that are living.
Cool. So we'll have you all back for a follow-up in a couple of years again. Next question is cool talk. Sounds like this is under next steps, but curious if you have any ideas on the differential effects of these sponges on restoration objectives like fisheries habitat, inverse diversity, water quality, or binding potential.
So we actually have a proposal that that's pending right now to do some of that work. So not to scale this up to sort of full implementation of restoration, but to actually increase the number of sponges that we have out there and look at things like diversity and abundance of sponges and how we distribute them, and then those knock-on effects, like depending on how many sponges we put out and how we distribute them, do we start to see recruitment of not only little baby sponges, but also other things that are attracted to that habitat, including fishes and invertebrates. And so we would look at, what those communities are like now, add these sponges, and then look at what those communities look like in 6 months, 12 months, 18 months. So yeah, so that actually is hopefully those objectives are, if that proposal comes through, be happening in the next year or two.
And I actually skipped a question that was, you started talking about permitting, but this is a question about seagrass beds. I also noticed there were some pictures of the seagrass growing in the background in a lot of the pictures. And it's basically, what were the permits you obtained for the work, and did short-term impacts to seagrasses become a discussion point?
Yes, that was a point. And so when we initially set up those nursery sites, we targeted places where there was no seagrass because the sponges really aren't going to impact the seagrass tremendously because eventually the bricks just kind of settle down into the sediments and really don't affect the seagrass tremendously. The little footprint of the brick isn't that great. And actually, what we'd like to do is put sponges in places where they will eventually connect to the bottom. So if we're putting them in places where there's seagrass, the sponges are never going to be able to actually connect to that hard bottom and attach. But in some places, what happened with these hurricanes over the past few years is they move sediments around and they move, and the aerial extent of the grasses will change and sort of infiltrate the hard bottom areas. And so that's one of the things that it's tough to necessarily account for, is the movement of sediments and essentially then the movement of the grasses into those areas. But the initial setup, that was a part of it, was avoiding seagrass habitat for those two reasons. We don't want to be in it and we don't want to impact it.
Cool. Awesome. Next question is light limitation an issue for the sponges, and do you think it explains variation between sites, times, or species?
Do you want to field this one, Aaron, or?
Yeah, it's a really good question. I don't know. I can only, speculate a little bit. I think turbidity might have some explanatory effect here. In that habitat, being as near shore as it is, you have a good wind, it seems like, and it'll, pick up a lot of sediment. And so there would be days when you'd be diving, and, the light penetration is probably pretty poor, really turbid water. So that, yeah, might for some species, impact some of those outcomes. I think if, maybe the yellow sponge does better in like less turbid water, or some species might do better a little bit further offshore where that's a little bit less impactful. But yeah, that's a good question.
So the sponges that we chose, other than the purple branching sponge, which I don't know if anybody knows its status, sponges tend to be either high microbial abundance sponges or low microbial abundance sponges. So they really kind of fall into those two classes. The other four species that we've used so far were all high, would are determined as high microbial abundance sponges. So they have lots of microbes in among their tissues that perform lots of different functions. One of the microbes that's very common in many of them is cyanobacteria, which is a nitrogen fixer, but also because it's cyanobacteria, it is photosynthetic. And so those species, if they have a strong reliance on those symbiotic cyanobacteria, probably need to be in higher light environments. And so that's part of what we're hoping to figure out if we get some funding to do these latitudinal experiments where we basically put out cuttings of the same species at different distances from shore and different depths. Not only would we get some of the salinity information as well, but we'd also hopefully get some light information from those sites.
Wonderful. And in our last minute, the last question we have time for is, did you determine an optimal size to transplant? They appear to be mature specimens.
So the ones that you see in all those images are sponges that have healed, and they look like little baby sponges of the originals. And I think I had one image of a sponge early on in the presentation that was a cutting, and they're just literally chunks that we use. Typically, we try and use pieces that fit on the sort of footprint of a brick. So maybe they overhang a little bit. Sponges are somewhat, the different species can be amorphous, but generally that we would cut them into pieces. We want to minimize the surface area of cut that we have as much as possible. So we don't really cut them like birthday cakes, right? So they just have a little bit of tissue along one edge and then a long slice where it's just exposed internal tissues. So we try and have as much of that sort of integument exposed to the outside water as possible. So yeah, I think most of the images you saw, they heal over relatively quickly and just look like little tiny clones of of the parent, quote unquote.
Wonderful. Well, it is 3:00, so unfortunately we are out of time. There were a couple questions in the chat we didn't get to, so sorry about that. But thank you all so much for your engagement and all the questions. And I'll send a reminder and registration link to everyone about our program for next month. But we have Dr. Ed Camp, who's going to be talking about how we know what anglers are targeting and fishing for and where they're coming from in our coastal waters. So that should be a fun and different talk. So thank you so much again, Aaron and Don, for your time. And we really appreciate all of you taking the time to be here with us.
Right. See you all
Thanks
later.
so much.
For having us.
See you next time.